This is lecture number 6. This lecture is on maps in the brain. So, even in the very first lecture we mentioned that there are these maps in the brain. So, information is often laid out in the form of 2 dimensional maps on brain surface. We talked about the somatosensory map or the touch map, how the surface of the body is mapped onto this somatosensory map, where nearby points on the body surface are mapped onto nearby points on the cortical surface. We also talked about the motor maps, where the entire body is controlled by these maps in the motor cortex. So, the left part of the brain controls the right part of the body. So, if you activate the nearby points in the motor map, you produce movement in the nearby points on the body. In this lecture we will talk about how these maps are formed, what are the mechanisms. It turns out that although the maps are pretty complex and they are found in many modalities, not just in touch and motor or movement, they can be explained using very simple mechanisms. Actually, there are only 2 rules by which you can explain a lot of this map phenomena, and that is what we will be looking at in this lecture. So, first of all, let us talk about what is a map. Right? If you look at the dictionary definition, it is a representation usually on a planar surface of a region of the earth or heavens. So, in fact, in this map this is a map of it's a world map, right, that was popular in Europe in the during the dark ages. So, because in those days they did not know much about the rest of the world, they knew that there is Europe and they knew only a part of the Africa, the northern parts of Africa. They knew that there is China and India somewhere towards the east, but right, and around and all around they thought there is this large world ocean. So, so let us talk about what are the features of any map. So, in the map it consists of a 1-to-1 correspondence between the real world and the map, and nearby regions in the map are mapped onto nearby points on the map surface. Map may or may not be drawn to scale, and also there can be local distortions depending upon your emphasis. Right, if you want to emphasize and expand certain parts, right, then you will introduce a local distortion. A good example of such a distortion, which is quite dramatic and even funny, is this map of the world from the 9th Avenue of New York. This was drawn by Saul Steinberg. So, New Yorkers generally they think no end of this city and they think that is the end of the world. So, in this picture you can see the 9th Avenue in the foreground, and behind it you see the 10th Avenue, and after that very quickly you see the Hudson River, and beyond that you see New Jersey, and you see various American states, cities like Las Vegas and states like Utah, and you can see Chicago right towards the right, and then beyond that you see Pacific Ocean, and beyond that you see the eastern world, China, and so on and so forth. So, the point is, in this extremely warped picture of the world, right, the 9th Street of New York, Manhattan occupies a big area, and the entire world is organized around it. So, this is a funny illustration of how maps can be very distorted. And interestingly, you see such distortions even in the brain maps for various reasons. So, brain maps consist of information laid out on 2D sheets of neurons. They can be cortical maps. In fact, the kind of maps we have encountered so far are mostly cortical maps, but they can be subcortical maps. There are many such maps in the brain, and very often these maps are interconnected in complex fashions. I think we once mentioned before in one of the introductory lectures, the first person who popularized these maps and did a lot of pioneering work in these maps is Wilder Penfield, a Canadian neurosurgeon, right, who is often called the neural cartographer or a neural map maker. So, he did his he started his career based on studying certain brain stimulation experiments. So, basically a lot of his surgeries were addressing the problem of epilepsy, where there is this abnormal activity which starts from a part of the brain and spreads to the entire brain, and often this source of this kind of activity is in the temporal lobe. So, so the point which is called epileptic focus. Now, this tissue which acts as a focus anatomically and structurally looks similar to the surrounding tissue. So, the only way you can expose the tissue is by by functional stimulation. So, Penfield would go and expose that part of the brain and stimulate it, right, to see if stimulation of the given spot gives rise to triggers the epileptic seizure. So, in the process he stimulated various parts of the brain and found that very often information is laid out or organized in the form of maps, and he would draw pictures like this, right. So, in this picture you can see some sensory maps, the auditory maps in the superior temporal area, then the motor maps, and and so on and so forth. So, more modern portrayals of the maps look something like this. So, on the left here you see the somatosensory cortex, and you see how the whole body surface is mapped on to the brain surface. Here the somatosensory cortex, as we have once mentioned before, is in the postcentral gyrus. So, here you see the hand is mapped onto on this region, and the entire face is mapped onto this region. And what is interesting is the trunk, which is a very big area, right, is only this small region, right, and so on and so forth. So, so it is a highly distorted map because the hand, which is actually has a very small area, is mapped onto a very large cortical surface, whereas trunk, which is actually a very large area, is mapped onto a very small part of the cortical surface, and so on. And also in the face, face occupies a big area here. Even within the face, the lips occupy a huge area, right, which is very interesting. So, then again on the motor side, you see the motor map on the right side. You see the face here in this area. So, motor map of the face involves movements of your jaw, your tongue, your lips, and other facial muscles. So, the hand also, even in the motor map, hand is allocated a very big area in the motor map. So, specifically as far as motor maps are concerned, although these maps look very clean and point to point maps from the body to the brain, on the motor side actually maps are not that clean, they are somewhat more fragmented, right, compared to the maps of the body surface on the side of somatosensory maps. So, people would made all these maps and after the early discoveries of Penfield, and then they thought that inside the brain there is some kind of a little man, right, which is which these maps embody, and this little man is receiving all the input from the external world and operating on them. So, they called this kind of imaginary little man homunculus. Homunculus means little man, and they would make little figurines of this kind of imaginary persons, right, and these figurines would decorate the tables of neuroscientists, and this kind of thinking about how maps work became very popular, but this is definitely an oversimplification of what the map means. We talked about the maps in the somatosensory area and the motor maps, but maps can be found in every other modality. So, for example, there are visual maps of the retinal image onto the visual cortex, right. Some of the earliest work on the visual maps in the brain came out of the work of a neurologist called Gordon Holmes, right. He is a British neurologist who worked on World War I victims. So, a lot of these soldiers who were wounded in the war came back with penetrating wounds produced by the shrapnel, right, of the war, and 25% of these wounds were head injuries, and part of the reason was the bad design of British helmets. So, in this figure you can see the British helmet on the top and the German helmet on the bottom. So, you can see very clearly that the German helmet is better designed as far as the protection it offers to the back of the head. So, you see that extended part of the helmet in the bottom, right, and that really covers the nape of your neck, and whereas on the British side in the British helmet you do not have that kind of an extension. So, therefore, a lot of these wounds, right, would affect the back of the head and injure the occipital lobe, which is where you have visual area. So, he would study these patients and look at how their vision is impaired because of the wounds that they have in the brain, right, because of shrapnel stuck in the visual cortex. And what he found is the shrapnel is very small, right, and often almost like a pinprick. There is a corresponding deficit in the visual field. So, they would just see a kind of a black dot or a black patch, a dark patch in the visual field. So, it is as though the input coming from that part of the image is not being processed by the brain, by the visual cortex. And he also noticed a very interesting relationship between the location of this black spot, which is called scotoma, and the location of the wound in the visual cortex. So, if you look at this picture, we have looked at the organization of visual system earlier in some of the earlier lectures. So, we know that the image now goes to the retina and image is formed on the retina. From there it goes to the thalamus, right, and from there it goes to the primary visual cortex in the occipital lobe here. So, what he has noticed is a wound produced in the lower part of the occipital lobe, right, then the corresponding scotoma was located in the upper part of the visual field. So, if the wound is in the lower part, especially below the occipital pole is basically this tip of this occipital lobe where there is a sharp extension. right. If the wound happens below that, then the deficit that you see, the scotoma, is located in the upper part of the visual field, and if the wound is in the left part of the brain, then scotoma was on the right. So, there is an interesting mapping between the external visual field and the location of the wound in the occipital lobe in the visual cortex. So, later on people studied this mapping between the retinal image and the visual field and the brain surface, and these maps are called retinotopic maps. To calculate these maps or to measure these maps, people use this kind of a interesting grid patterns. So, in this grid pattern you can see a bunch of concentric circles. So, there are two dimensions with which you can characterize this a given represent any point on these concentric circles. So, there is a radius here which is called eccentricity or denoted by epsilon, and then the angle around that goes around the circle which is called azimuth. Okay, so, in simple high school mathematics terminology, the eccentricity is like the radius and the azimuth is like the angle. So, this is like polar coordinates of the visual field. So, this kind of a black and white picture is flashed in front of the animal. So, some of these recordings were taken from monkeys. Monkeys have very rich visual system which is quite close to human visual system. So, very often monkeys or primates that are used as experimental animals for studying the visual system. So, this kind of bullseye pattern was displayed on the screen, and the corresponding activity in the primary visual cortex or V1 is measured using radioactive glucose. So, basically what they do is they inject radioactive glucose into the bloodstream of the animal. So, we know that neurons need glucose from the bloodstream to run their firing activity. So, this radioactive glucose is picked up by active neurons, and therefore, wherever neurons are active, they take this tracer, this radioactive glucose, which can be seen in autoradiograph, right. So, so that radiograph looks something like this. So, on the this image on the left side. So, the bar patterns that you see, the grid like intersecting lines that you see in this radiograph indicate where the neurons are activated, because these are the points in the brain surface where neurons have taken up the radioactive glucose. So, what is interesting is the image shown to the animal and the pattern of firing of neurons in the visual cortex, they do not look very similar. Now, you can see a grid like pattern, but it does not look like a kind of a carbon copy of the image that you see in the external world. So, in fact, what this experiment tells you is that the activity pattern of neurons in the visual cortex, right, it does not duplicate what you see out there in the real world. It really transforms it in a very profound ways, and then this transformation gets more and more complicated as you go to higher visual areas, which we will which we cannot discuss at this point. So, so this grid like pattern can be mathematically represented in a simpler graph like this, what you see on the right side. So, what you see here is the eccentricity, which is radius in the input space, in the visual space, turns out to be some kind of a x coordinate, and the azimuth, right, which is like the theta or the angle in the polar coordinates, happens to be the y coordinate in this space. So, what is radius in the polar becomes x axis in the cortex, and what is angle theta in the polar coordinates becomes y axis in this space, in the cortical space. So, this can be expressed using a very interesting formula. So, here z is a complex number, and those of you who had studied complex algebra in your high school can recollect some of this information about complex numbers. So, if z is a complex number here defined in terms of the eccentricity E epsilon and the azimuth a. So, I is the imaginary number, purely imaginary number, right, and so this formula defines a quantity called z, and z is a complex number which defines a point on the visuals in the visual space. And given that point, this capital Z is a point on the cortical space, which you can see like these are points on the cortical space. So, the visual space to cortical space mapping can be defined by this formula lambda ln z. So, ln here is the natural logarithm, but it is a complex ln function. So, it is a complex transformation from the visual space to cortical space, and experimentally people have found that this formula with a small with small variation, right, can explain the retinotopic maps of a large number of species. So, which is very interesting. So, this is one more example how mathematics, and that too such an esoteric mathematics like complex algebra, can be used to explain something so deeply biological. So, this is an interesting example of that. So, now a retinotopic map basically tells you how the visual space is mapped onto the cortical space in the occipital lobe in the primary visual cortex. Now, let us look at the response of single neurons and how what kind of maps they form, right, about the visual input. So, Hubel and Wiesel, no, these two people were working in Harvard in the 60s on the response of visual cortical neurons, and what they found is so neurons in V1 respond to bars and edges. So, okay, some of these experiments goes like this. So, you have a pattern or a picture that you present on a screen, right, and record from various parts of the visual cortex by poking the neurons with microelectrodes. So, the experimental typically tries to figure out what kind of a pattern shown on the screen produces a strong response in the neuron from which you are recording. For example, if you show a flash of light or a dot on the screen, if the neuron responds strongly to that input, it means it recognizes that pattern and it is tuned to that pattern. The idea of tuning is very important, and we will return to that concept later on in this class. So, they did that, they flash different kinds of patterns and recorded from lots of neurons and trying to see what they respond to. And what was known earlier before these kinds of studies based on recordings from the retina and the thalamus or the lateral geniculate nucleus is that neurons in these lower stages of visual system respond only to dot patterns. So, that means you could, if you have a white dot on a black background or a black dot on a white background, typically these are the kinds of patterns or stimuli that elicit a strong response in neurons in the retina and in the lateral geniculate nucleus. But the same kind of stimuli fail to elicit any interesting or significant responses in the neurons of visual cortex. So, for example, if you look at this picture, if you showed little light spot, right. So, this is the receptive field of a neuron. So, I am recording from this neuron, and so this circle indicates where all you can give an input so as to produce a response in this neuron. So, if you give an input outside that circle, it will not be able to reach that neuron. So, whatever you do, you have to do within that little circle. So, any kind of a dot like pattern, a symmetric circular stimulus fail to produce much response right in the neuron, right. You could increase the size of the circle and all that, not much response. On the other hand, when they showed a kind of a bar like pattern, a linear pattern, right, that showed in significant response, particularly when the bar has a certain orientation. So, on the left you can see that when they showed a horizontal bar, no response, right, when they showed a bar at an angle, no response, and as the bar became more and more vertical, right, suddenly this, so there is a lot of response. So, these lines here indicate the strain of action potentials emitted by this neuron. So, again when the bar is rotated further, right, as it is moved towards horizontal position, there is not much response. So, that means this neuron is responding to oriented to bars first of all and not to dots, and further it is tuned to bars with certain orientation. So, each neuron here is tuned to certain oriented bar. So, so that was what is interesting about neurons of V1 compared to neurons in the lower stages. And they also found some neurons responding to moving bars. So, for example, there might be a neuron which responds to a vertical bar when it is not moving, immobile, and there are neurons which respond to a vertical bar when it moves to the right, and similarly there are neurons which respond to a vertical bar when it moves to the left. So, there are neurons that respond to all sorts of linear stimuli and not to circular or symmetric stimuli like dots or discs. Then there are also different neurons respond to different kinds of orientations, and within a small cortical area of about 1 millimeter, right, they found neurons which respond to all sorts of orientations. And further on here they found different subclasses of cells which respond to orientation in slightly different ways. There are these simple cells, right, which respond to oriented bar only if the bar is located in the center. If the bar is located off center, the response is somewhat weaker. And then there are complex cells, right, which respond when the bar is anywhere within that receptive field. So, the orientation of the bar should be correct, right, that is right for that neuron, but it can be anywhere within the receptive field. Then there are end stopping cells, right, where the stimulus should be present in the receptive field, but the line very often just stops somewhere in the center of that circle. So, it does not go through the entire receptor field. These are called end stopping cells. So, they found different types of neurons which respond to different types of lines with specific orientation. Then they also found neurons which respond to edges. So, the example that we have seen before just a moment ago are examples of lines, neurons which respond to lines. So, a line is basically like a bar with some, say, with certain orientation, and there is a contrast between the part of the line and its and its surround. So, in this case I have a dark line with a white surround, in this case I have a white line with a dark surround. Okay, both of them are like lines or bars, and there are neurons which respond to these kinds of oriented bars. There are also neurons which respond to edges. So, edge again is a linear boundary between two regions where there is a contrast. So, in this case in the upper part of the edge there it is dark, and lower part it is white, and in this case in the upper part it is white, and in lower part it is black. So, these kinds of all these kinds of edges or bars, right, typically trigger responses in the neurons of V1. Okay, so now the experimentals also recorded from neurons at various depths inside the cortex. So, we know that cortex is a thick sheet of cells, is a slab of cells, and within this slab there are 6 layers. We talked about this before, and they used a microelectrode and penetrated deeper and deeper levels of this cortex and recorded from neurons. When they did that, they found that neurons at various depths responded to the same orientation. So, which is what you see here in this nice schematic. You see the neurons at various depths respond to the same orientation. At some points you have neurons which are not orientation sensitive. So, for example, here they do not respond to any particular angle, but otherwise when they respond to an orientation it is often to the same angle if you measured from various depths at the same position in the cortex. But what happens if you recorded not along the depth, but along parallel to the surface of the cortex. So, in this in the schematic on the right, they recorded from various neurons, right, at various locations, and they proceeded along a straight line, right, parallel to the cortical surface at certain fixed depth. When they did that, they found that the different neurons responded to different orientations. So, here the orientation sensitivity seems to be changing along the length, along the length of this path of the electrode, and but this variation is quite ordered. So, for example, at this extreme end you have a neuron which responds to horizontal bars. As you move further and further along this path of the electrode, you find that the orientation tuning of this neuron also rotates quite gradually, and somewhere here in the center you have a neuron that is here which responds to a vertical bar. As you went further down, somewhere here you have a neuron which responds to a horizontal bar. So, you have come full circle. So, if you go further down, you will find somewhere later, right, neuron responding to a small orientation, a line with a small orientation. So, there is some kind of periodicity here because the angle is a periodic quantity. So, if you kept on measuring the orientation tuning of various neurons along a straight line in the cortical surface, you will find that the same angle from 0 to 180 degrees repeats, right, all along the way. But now the question becomes very interesting, because cortical surface is a 2-dimensional surface, it is not a it is not a straight line. So, if you went in different directions, how does the orientation sensitivity change? How does this tuning angle of orientation, right, change from neuron to neuron as you went around the cortical surface of the primary visual cortex? So, people measured this kind of tuning, and these are called orientation maps. These are 2-dimensional maps which tell you what is the orientation angle, right, to which a given neuron responds at a given point on this 2-dimensional surface. Now, since they are maps, you have to make 2D pictures of them, and here is an interesting color chart or a color map that indicates orientation of various neurons in this primary visual cortex. So, each color indicates a certain orientation, and there is also, you can see these short white lines, right, which indicates also indicates orientation. So, for example, the red color indicates horizontal lines. So, neurons here respond to horizontal lines. So, similarly, the green color indicates vertical orientation, that means neurons in this part of the map respond to vertical lines, right, and so on and so forth. So, yellow indicates orientation of about 45 degrees, right, and so on and so forth. So, you see how different neurons respond to different kind of orientation in this map, and there is a kind of a gradual variation of orientation from point to point as you go over this over this cortical surface. Now, there is a different kind of a map which is also superimposed in the same space of primary visual cortex, right. To understand these maps, we should understand how the visual space is mapped onto the cortical surface. So, we have seen this once before. So, each eye receives input from both the left part of the visual field and right visual field, and if you look at the right eye, the right part of the eye, which is called the temporal side, right, receives input from the left visual field, and the left part of the eye receives input from the right visual field. Also similarly, on the for the left eye, its right part receives input from the left visual field, and its left part, which is called the temporal side, receives input from the right visual field. Now, you see that the way the information is propagated or projected backwards to LGN and further down to the primary visual cortex is very interesting. So, the information from the temporal part of the right eye and the nasal part of the left eye, both of them correspond to the left visual field. Both are sent to the same side of the brain, which is the right brain. So, the information about the left visual field is captured partly in the left eye and partly in the right eye, but both these chunks of information are sent via complicated wiring patterns to the same part, or combined and sent to the same part of the brain, which is the opposite side, to the right brain. So, similarly, information about the right visual field is captured partly by the left eye and partly by the right eye, but both these chunks of information are combined and sent right to the left part of the brain, to the left primary visual cortex. Okay, so, very interesting kind of a regrouping of information happens right through this wiring pattern from the eyes to the primary visual cortex. So, that means a given part of the primary visual cortex receives input right partly from the right eye and partly from the right eye, but it receives input only from the left visual field. Okay. So, now the thing is, within the primary visual cortex on the right side, you will have neurons which respond to inputs coming from the left eye, and there are neurons which respond to inputs coming from the right eye. Now, the question is, how are they organized spatially? So, if you can color the neurons as with black and white, right. Black is neurons which respond to left eye, and white is neurons which respond to right eye, right. How are these two neurons organized spatially on the visual cortex? So, what they found is, here also they found they are organized in nice periodic fashion. So, for example, in the cortex, if you record from layer 4, right, and you will find, let us say, neurons which respond to left eye here, right, over this whole stretch. And after that, again you will find a stretch where neurons respond to right eye, and so on and so forth. So, you have a simple repeating periodic pattern of neurons which respond to right eye and left eye. So, this map is still one dimensional, but if since cortical surface is two dimensional, if you look at the same maps in two dimensions, right, it looks more complicated. So, in the image here shows a map of the neurons which respond to right eye and left eye. So, in this case you can say that the black here corresponds to neurons responding to say right eye, then the white corresponds to neurons which respond to left eye. So, these kinds of patterns are measured or produced by injecting a tracer, a radioactive tracer like right tritium amino acid tracer. They inject this tracer into one eye, and the signal goes through the pathways, the optic nerve, and crosses the lateral geniculate nucleus and reaches the visual cortex, and activates only the neurons which receive input from that neuron in the retina. Okay, so, this is a map of one eye and bunch of neurons which receive from that eye. So, what is missed out in the map has to be neurons that receive input from the remaining eye, the other eye. Okay, so, you see very interesting zebra like patterns, and these patterns are called ocular dominance maps or ocular dominance columns. Okay, and also these maps are not fixed. So, for example, if they are driven by the external input, and so if you change the external input, your visual stimulus, then these maps reorganize and change. So, for example, in this experiment which was done with monocularly deprived monkey. So, they take this monkey at an age of about 2 weeks to 18 months, which is what is called a critical period. This is a period in which the brain of the baby monkey undergoes lots of changes in the map organization. But after that, maps stabilize, and there is not much, you do not see much change after once the critical period is passed. So, during the critical period, they molecularly deprived, that is they just shut off one eye by suturing the eyelids, so that that eye cannot receive input receive light. When they did that, they found that the maps form in such a way that neurons predominantly respond to the opposite or intact eye. So, there are very few neurons left to respond to the eye which is sutured. So, because of that, even though the sutures were removed at the end of this period, right, there are not many neurons left to respond to input coming from the light from the. So, this let us say the right eye is sutured. So, not many neurons left to respond input from the right eye. So, it is as though the brain is blind to the right eye. So, we have a very interesting situation here. The right eye is intact, I mean right now the right eye is open, but the maps, right, have formed such that the neurons which respond to the right eye, right, are very few. Therefore, it is as the brain cannot receive input from the right eye. So, this is a situation of what is called cortical blindness. Normally, blindness is associated with eyes, something is wrong with the eyes, but here the eyes are fine, but the blindness is in the cortex because the maps have not formed, or maps are totally skewed to one eye and the other eye is functionally blind. So, in fact, there is a interesting so this raises lots of implications for blind people, right. So, because the condition for seeing is not just intact eyes, but intact maps, the visual maps in the visual cortex.